My first talk now is about immunogenicity. This is the definition of immunogenicity. Everything that has something to do with an unwanted immune response against itself or to related proteins or to induce immunologically related adverse events. So, and the main criterion is the formation of anti-drug antibodies. So this is the definition. Why immunogenicity is important? We got this from the discussion from this morning, because every protein-based biologics have an immunogenic potential that can cause anaphylactic problems. Via these antibodies, they can have an impact on efficacy, on binding affinity, and the regulatory agencies, they need to figure it out in their recommendations to get data from immunogenicity problems or from immunogenicity itself out of the whole development process. Well, this is just to illustrate the considerations for immunogenicity assessment during the product development, and it has to be done in the early situation, in the clinical development, and of course, I think this is of highly importance after approval. And we talked about registries. I don't think we will see these switching studies Paul illustrated, and I hope that we get sort of registries, but I do 2 registries myself, and I know about the quality of data that is included in these registries. This is really very variable regarding what kind of data are implemented in these data sets. What are ADAs? I think you know what ADAs are, anti-drug antibodies. This has something to do with our immune system. What happens after ADA has been performed? Well, we get different reactions, can lead in destruction of macrophages and can cause the destruction by cells via macrophages, and so on. But all in all, it will lead to different anaphylactic reactions, and they can be mediated by different immunoglobulins like IgE for the immediate reaction, IgG or IgM for more delayed reaction, and this can take 1 to up to 3 weeks or a couple of days. So this is everything we know from our side effects. Different medicinal products can generate, and this is something we have to document, and of course we have to correlate with the different biosimilar products. All in all, we can define non-neutralizing antibodies and neutralizing antibodies, but both types of antibodies can affect the efficacy of our medicinal products. And factors affecting immunogenicity can be drug-related, can be patient-related, and can be treatment-related, and all these different factors can influence immunogenicity. And when I talked about a sensitive patient population, this has something to do with immunogenicity. And when I talked about different lines of treatment and changing the cancer cells, this has something to do with immunogenicity. And of course, the mechanism of action, the route of administration, frequency, duration of therapy, and so on. This is what I mentioned before about Trastuzumab. It has, like different other antibodies, it has different characteristics and different functions. It can induce ADCC, can block the receptor, it can prevent cleavage and shedding of the receptor, because if you don't have an extracellular domain, of course, there's no binding anymore. So therefore, we have to avoid this, and it increases HER2 receptor endocytosis. And these are different mode of actions, and all of this has to be proven in the biosimilar development process. And this is what I think of a mode of action. It's not that easy, it's more complex. Product-specific factors can affect immunogenicity, and protein glycosylation was mentioned before. Yes, it is valuable if you have mannose or fructose or different other glycans implemented in the biochemical structure. And if you have, let's say, a bit more fucose, then maybe it will affect the binding affinity of your product. We have patient-related factors, as mentioned before, genetic factors, factors that has something to do with the immune system, the function of the patient, and immunogenicity, therefore, needs to be evaluated, not going to go back in this area of PK and P data generating data. It has to be collected via or in the clinical situation in clinical trials as well. Impact of immune response on safety and efficacy. To make a long story short, I think the cartoon down there illustrates quite nicely that we have a big range between no apparent effect and a fatal situation, no event, serious adverse events influenced by neutralizing or non-neutralizing antibodies. So we have between nothing and loss of efficacy, this is what I want to tell you, a wide range of influences we have to take under consideration, and we have to document and to collect in a post-marketing phase. And this flowchart just illustrates that neutralizing antibodies, so say antibodies that bind to active site, antibodies that affect conformation of active site, and non-neutralizing antibodies that do not affect the active site, both have influences because they can induce aggregation, can alter pharmacokinetics, and of course have an impact on efficacy, potential impact on safety. This is just a summary what we'd have to take under consideration when we think of an impact of immunogenicity on safety on the left side and efficacy on the right side. I think I told you this already, we can skip this. Then this morning we had this question how to detect anti-drug antibodies, and of course there are different assays that can be done, and every assay has to be confirmed. That means I have an ADA screening assay, different assays existing, then I have to confirm this, and then I have to make an ADA characterization assay if I have a positive result to detect neutralizing antibodies in then the following neutralizing antibody assay. And there are different assays, and well, my years in the laboratory a couple of years ago, but we use different kinds of tests, and of course you have pros and cons, and there's not a perfect test in the end, and it depends on who you talk to, and then you get more pros and more cons, but in the end there's not a perfect test to screen anti-drug antibodies. There are some clinical examples, and I think this is important, that we have limited data out of the biosimilar somatropin area and the EPO area, and there is some data existing that for biosimilar somatropin, up to 60% of patients developed anti-growth hormone antibodies. For EPO, some patients got really severe toxicity. In one case, a patient developed pure red cell aplasia. So it has something to do with the data I collect afterwards. So to reproduce myself and to repeat myself, and so on. Well, the regulatory perspectives, of course, the FDA and the EMA, they say immunogenicity is a key issue, and for an approval of biosimilars, both EMA and FDA require comparative immunogenicity assessments with a reference product in clinical studies. I will skip this, and there are different methodologies. Of course, it's always more expensive to have a two-assay approach to compare the biosimilar compound and the reference product better, of course, than just to do your immunogenicity assays on the reference or the biosimilar compound. So we have more pros in a clinical, for the clinical point of view, and less pros for the clinical point of view, but for the costs, of course, because costs are increasing if I use the two-assay approach. There was a letter from the ASCO, from the President Dan Hayes, to the FDA, figuring out that the FDA has to react, and this is too busy really, and has something to do with please clarify interchangeability. Paul already mentioned in his talk, please clarify switching and alternating studies. We have to have recommendations, detailed recommendations for these issues. Some more, and what I want to focus in, that they say we have to have post-marketing studies or a clinical trial to evaluate certainty, safety, risk, and of course this includes immunogenicity. To conclude, all protein-based biologics have an immunogenic potential. This is what you know. Anti-drug antibodies are produced by B cells. They can affect the therapeutic effect, and therefore immunogenicity is a safety concern for biologics, including biosimilars and biosimilar antibodies. We have different influences on the immunogenicity by product, patient, treatment-related factors, and consequences are, of course, adverse events in a range of no really event up to a fatal event, and long-term immunogenicity has to be monitored during post-approval surveillance in a real-world scenario, and this is very important. I think I will go, in a matter of time, to the next talk, and if we have time left, then we can talk to each other. Otherwise, we have to talk in the lunch break, because I really have to move forward. And the next talk is about issues concerning naming and pharmacovigilance. Yes, it is important for biosimilars to be properly named and identified, and it's good that this is the last talk, because everything we talked about, Professor de Klerk and myself, has always something to do that in the end you can identify as the prescriber what you give to your patient, because otherwise you cannot correlate all these factors of efficacy and safety in the end. And I really loved your example, Paul, with these switching studies. In the end, you really get totally mixed up by having, 6, 7 choices, and then you don't know what kind of adverse events belongs to the biosimilar compound. So naming of biosimilars, we have recommendations by the EMA and by the FDA, but in the end, it's good pharmacovigilance is important for detection of rare and unexpected safety issues, and we have to have unique names for a proper distinguishing of the different products. And this is really a challenge when you see what the current naming policy is over the last decades, and you see that the number of EPOs, for example, increased, and if you prescribe by INN, then you have Epoetin, and you can distinguish this by Epoetin theta, so a Greek letter, Epoetin zeta, alpha, and so on, but then you have different Epoetins alpha and different Epoetins zeta, and then you get mixed up when you prescribe just an Epoetin alpha for my patient. Therefore, if you have to report adverse events, then you have to know the brand name in the end, because prescription by INN, if I, as a physician, write just down in Epoetin, then the pharmacist dispenses what is available and what is less expensive, and my hospital pharmacist will do this. But if I write down a brand name, then he is forced to communicate with me if it's not available, the compound I prescribed. Therefore, we as prescribers must be able to track accurately which particular biologic was given to a patient to allow, in the end, an adequate pharmacovigilance. I will skip this, and of course there are directives from different years, but all in all, the EMA guideline on pharmacovigilance practice for biological medicine from the year 2016 says we have to capture the brand name and the batch number. Brand name and batch number. This is what I told you already. And in a real-world scenario, and this is what I think is important, and this is a study from the Netherlands on traceability of biologics. Here, more than 1,500 ADR reports were analyzed, and overall, in just 76% of these ADR reports, the brand name was stated. And 93 of the 80 reports for opinions had a brand name, but 0 of the 80 reports had a batch number included. And when I think of my systemic treatment ward, we have a year around 5,500 cycles of chemotherapy in my department. No one writes down the batch number, and no one writes down the brand number, which is, we say, well, this is the INN, and now we have to change, really. And this is, I think, really hard to train, but in the end we have to do this. There are different possibilities. One I figured out already, brand name plus INN plus the Greek letter, or brand name plus INN plus biosimilar and a number. For example, filgrastim, then you have filgrastim, biosimilar number 3, number 4, and so on. And what the FDA recommended, the brand name and the INN, and then a 4-letter suffix. And there's an example, sarxofilgrastim, SNDZ, and this comes from Sandoz, but SNDZ doesn't say it comes from Sandoz, so this is by accident, and there's a discussion going on. Do we have to use 4-letter suffixes that can more identify the product, or is it just by accident? These are different examples, and there you see adalimumab, for example, ATTO has nothing to do with the company, it's just by accident, and infliximab A, B, D, A. So, well, maybe it's better to recommend to use these suffixes that has something to do with a company or makes it easier to identify. So this is a discussion that is going on, random or not random, is this meaningful? Short conclusion, I will skip because I have told you already, and the labeling of biosimilars, we have different approaches. A generic approach, label to be an identical copy for the reference product. Then we have an innovative product approach. This is the label to include information on the biosimilar, and then we have a combined and I think more transparent approach to combine information on both the biosimilar and the reference product. And this is an overview what kind of approaches are used in the US, in the EU, Health Canada, and so on. And in the EU we have a generic approach, and that means that the label, and this is recommended by the EMA, has to include a biosimilar statement, but just a statement, and the rest, so the summary of the product characteristics belongs to the reference product. I think I have an example here. So this is one example for Remsima, for example. So the label is totally the same. And then in section 5.1, and there is written Remsima is a biosimilar medicinal product, details, information, and so on. But there's no information about the clinical data. So what kind of study was done? It's just biosimilar, and the data set that exists is from the reference product, and the data set we have for the biosimilar compound is just to show biosimilarity. And this is something that is really strange to me, but this is the biosimilar word in the end, that we have to show biosimilar similarity, and therefore the label just includes one sentence, this is a biosimilar product. More transparent would be combined labeling approach, like in Canada, that we distinguish between data from the biosimilar and from the reference product regarding adverse reaction, regarding the data from clinical trials. So therefore, there's a bit more transparency, and for me as a physician who is interested in clinical data and of course in safety issues, it would be a better label than the generic approach that is done in the EU. Of course, stakeholders recommend that a combined, more transparent labeling approach is preferred. And this is another publication where a survey of 210 physicians across 7 different European countries were asked, what do you want, what do you prefer? And of course, a large majority preferred the modification of the label for biosimilars in terms of a combined approach to get more transparency. My last slide to summary and conclude. Labeling approaches for biosimilar differ globally. The combined labeling approach is what I think more transparent in terms of the source of clinical data, extrapolation, safety profiles, what I'm interested in as a prescriber. The majority of stakeholders is the same opinion, and physicians would prefer the label, what I said, with a combined approach. And that's it. And yeah, thanks for your attention.